as hopefully my introduction maybe kind of laid the groundwork for, I'm kind of maybe a non-traditional researcher in the diabetes space, right? I'm not an immunologist, I'm not an endocrinologist. I'm an engineer by training, and I kind of consider myself an imaging scientist. And my goal today is to hopefully convince you that there's a role for imaging in diabetes to help us advance our understanding of diabetes and possibly form a useful clinical tool for monitoring or detecting or diagnosing diabetes. So that's kind of my first goal. My second one is to convey the importance of machine learning for analyzing all this medical imaging. I think it's really shown its use in other medical imaging applications, and I think that the application to diabetes will likewise really benefit from deep learning techniques. And then at the end of the talk, I'll talk a little bit about my dkNET pilot project and how I'm using dkNET and NIDDK central repository resources to hopefully to move this field forward. All right. So we typically think of type 1 diabetes as kind of a pancreas-derived disease, but we don't actually assay the pancreas when we're diagnosing or monitoring type 1 diabetes, right? So we typically look at blood biomarkers. But when we think of the kind of natural history of type 1 diabetes, it's probably starting primarily in the pancreas, where the immune system, it has some triggering event which sets off some autoimmune process. And over time, individuals who develop type 1 diabetes are losing beta cells. And this time scale can be long. It can be months. It can possibly be years. We don't really know because we don't actually have a way of imaging the beta cells directly. But typically when someone presents to the clinic with, high blood glucose values and is diagnosed with diabetes, we think that they've probably been losing their initial beta cell mass for a long time, and they've probably lost the majority of that initial beta cell mass. So we think that they've probably lost the majority, probably at least 50%, possibly as high as 90% of those initial beta cells before they kind of reach this threshold and have clinical diabetes. And that's partially because we think beta cells have this really high compensatory mechanisms where initially, even if you're losing beta cells, the beta cells that are left there are just kind of working harder, but eventually they too tire out and you just kind of end up eventually losing what residual beta cell mass there is. So when I started kind of thinking about this project, I was an imaging scientist and was interested in kind of different imaging techniques, but then also applying them to different disease states. And one of the things that really interested me in this project is that there aren't a whole lot of imaging people who think about diabetes, right? There's a lot of imaging people who think about the brain, people who work with psychologists who try to figure out the way the brain works, people who work with neurologists and try to image stroke, MS, other neurological diseases. There are a lot of imaging people like me who work with cancer and are looking at ways to detect tumors, looking at ways to detect a successful treatment of a tumor. There aren't a whole lot of people who are looking at diabetes. So when an endocrinologist approached the group I was working with and said, hey, I have this interesting imaging application where it would be really useful to image beta cell mass. And he showed me a model, probably very similar to the one here, kind of conveying to me that it would be a really useful clinical tool to be able to monitor and image beta cell mass. And so when I was in graduate school, we made a transgenic mouse model where we expressed the luciferase reporter gene, so the same gene that makes fireflies glow. We expressed that under the insulin promoter in these transgenic mice, and then using these really sensitive cameras that could detect really small amounts of light, we could actually image the light coming out of the pancreas of these mice. So I thought this was really kind of cool technology, right? It's kind of pushing the limits of genetic engineering, kind of instrumentation where we're using these really powerful cameras that initially we actually had to pour liquid nitrogen over them to get them sensitive enough to detect these small amounts of light. And we thought this was a pretty cool preclinical model. We crossed it with the NOD model of type 1 diabetes. We crossed it with the DB-DB and OB-OB models of type 2 diabetes. We transplanted these glowing islets and we could detect them and image how they survived after they were transplanted. Looked at different therapies and whether those could induce beta cell proliferation. And we thought, hey, this is a pretty cool preclinical tool. But of course, one of my co-mentors, who was an endocrinologist, said, well, this is great. It can allow us to answer a lot of research questions. But for a clinical tool, we can't make a transgenic human. Even if we could, the human abdomen is just too thick. We couldn't image light emitting from a human pancreas. So we really need to move towards imaging modalities that are clinically translatable. And so I started working in what's kind of broadly known as the molecular imaging or nuclear imaging space. And the concept of kind of nuclear imaging, like a PET image or a SPECT image, I'm sure you're familiar with, is taking a radioactive molecule and targeting it towards something. So typically the kind of common PET ligand that's used in the clinic right now is FDG, or fluorodeoxyglucose. That's a radioactive glucose marker, and it's really useful because we can track where glucose goes in the body. So for instance, FDG goes to tumors because they're very glucose avid, and we can detect that glycolytic activity that's occurring in tumors much more strongly than the surrounding tissue. So we began looking at different radioactive tracers that might target the beta cells, or maybe more generally the pancreatic islet cells. And we try to take advantage of the fact that these islet cells are kind of neuroendocrine cells, right? Reflecting that they have endocrine properties, but they have some properties very in common with neural cells as well. So we attempted to use a number of different radio tracers that are commonly used for neural imaging. So they're used to look at processes occurring primarily in the brain. And we tried to repurpose those to image the beta cells. And what we found is that, we could see these radio tracers going into the mouse. We could see them getting uptaken in the central nervous system. We would see the striatum of these animals light up. So we knew the tracers were going to those neural cells, but we didn't really see them going to the islet cells or even to the pancreas. So our kind of forays into this field were largely unsuccessful. That said, there are a number of groups currently still developing these techniques, and some of them are starting to show some promise. So there is the possibility that in the next, decade, 2 decades, there might be a tool for imaging beta cells non-invasively in people. And I should note it's a really difficult problem, right? Because beta cells are small, they're scattered throughout the pancreas. We oftentimes try to make analogies to imaging a tumor, but when we image a tumor, we think we're doing a pretty good job if we're imaging a tumor that's 1 centimeter in diameter. Beta cells and islets are much, much tinier than that. So it's a really difficult problem from an imaging physics perspective. The other limitation is that these nuclear imaging techniques require us to inject a radioactive material. And certainly if we're thinking about doing this repeatedly, if we're thinking about doing this into a juvenile and adolescent population, as we typically see in type 1 diabetes, this gives us a lot of pause. So after these kind of forays into optical imaging and then nuclear imaging, I moved into MRI. And one of the nice things about MRI is it doesn't require any radiation. It's not like an X-ray where we're shining these X-rays that can cause these ionizing radiation and affect the DNA, causing mutations and possibly cancer. MRI doesn't do that. MRI is just a big magnet. As long as you don't have any metallic implants in your body, MRI is safe. We can do it in kids. We can do it repeatedly in kids, and we've done that now. So we've imaged a number of kids who have type 1 diabetes, a number of kids who don't have or do have type 1 diabetes, pardon me, and we see some marked differences between them. And I should note that the idea that the pancreas is smaller in type 1 diabetes is not novel. There's actually a Belgian pathologist who was looking at autopsy samples 70 years ago now, and he found that the pancreas of individuals who died from type 1 diabetes tended to be smaller than the pancreas of individuals who died from other causes. And it was markedly smaller. It's smaller by 30 to 40%. Now that we have these non-invasive imaging techniques where we can actually image the pancreas in vivo, accurately measure how big it is, we can use MRI to make these measurements non-invasively in people. And I've shown an example image from a control individual. This is a 10-year-old boy. He weighed 64 pounds over on the left. Kind of to orient you if you're not used to looking at these kind of images, these are axial slices. So imagine kind of the magician trick of slicing someone in half. That's kind of the view we're looking at here. The bottom of the image is the spine. You can see the liver over here, kind of on the left-hand quadrant. You can see this individual's stomach. You can see this 10-year-old boy just had lunch here. Here's his stomach, and it's mostly filled with food. He's laying on his back, so it's kind of settling down at the bottom of his stomach. Here's his empty stomach. we're really interested in this kind of bright looking organ right here. So this is the pancreas. And we typically think that in a kind of a healthy individual, it's roughly kind of a fattened banana shape, is our very unscientific way of describing it. And we go through all the slices of this boy's abdomen and sum up how much pancreas tissue is. We can see that his pancreas volume is about 27 cubic centimeters, or 27 milliliters. Conversely, if we look at the pancreas of a 10-year-old boy who has type 1 diabetes, as a 10-year-old boy, he weighed the exact same amount. He'd just been diagnosed with type 1 diabetes about a month prior. So he was newly diagnosed with type 1 diabetes. Again, I've outlined his pancreas in red, and I think it's readily apparent that his pancreas is much smaller. So we kind of talk about this fattened banana shape in the control pancreas. In individuals with type 1 diabetes, it tends to be this kind of compressed shape, and it especially seems like it's kind of swished through the body of the pancreas. And kind of incidentally, we work with a radiologist who's blinded to the kind of status of these study participants. When we show her the images, she doesn't know if somebody has type 1 diabetes or does not. But she said, incidentally, oftentimes when I look at these and I see a pancreas that looks like this kind of very thin, sort of not like a fattened banana, but kind of this squished banana shape, she said, that's someone I think probably is somebody who has type 1 diabetes. And we kind of informally tested her on this kind of thought. And, there's some truth to that. And so I'll come back to that shape analysis kind of at a later point in this talk. But just like the boy on the left, the control individual, where we summed up his pancreas volume, we can do the same for somebody with type 1 diabetes. And you can see that the pancreas size is much smaller. So same age, same weight, but the pancreas size is almost half as much, right? It's only about 14 milliliters versus 27 milliliters for this boy on the left. So pancreas size is markedly smaller in these individuals who have new onset type 1 diabetes. Now, I'm not going to talk much about type 2 diabetes, at least at the beginning of this talk, but similar changes have been found in individuals with type 2 diabetes. The decline in pancreas size might be a little smaller in people with type 2 diabetes, but there does seem to be also a decline in pancreas size in type 2. Now here's our cohort of individuals with type 1 diabetes. We've now imaged 56 control individuals without type 1 and 49 with recent onset type 1 diabetes. So these are all individuals who were diagnosed in the previous 100 days. On average, we're seeing them about 2 months after they're diagnosed. And you can see on average, pancreas size is smaller in those people with type 1 diabetes. Now we've imaged individuals across a wide range of ages, some who are kids. We image kids as young as 8 and some who are adults. Actually, before I get to that, I'm going to talk about kind of give you some context for these size changes, just to give you a sense of kind of what a 25 milliliter pancreas size might look like. So, a volume of Coke, a can of Coke is 355 milliliters. On average, the pancreas size in control individuals is about 50 milliliters. So think of that as about a seventh of a can of Coke in size. Pancreas size in somebody with type 1 diabetes is about 40% smaller, so it's 30 milliliters. And when we think about the islet volume of the pancreas, we think that islets or beta cells are 1 to 2% of the pancreas. And so the islet volume of this control individual would really just be a tiny sliver, right? So what this means is that there's some exocrine involvement in type 1 diabetes pathogenesis. So not only is this autoimmune process killing off the beta cells, but for some reason, you're also losing exocrine tissue mass. And we and a number of other groups are thinking about this and trying to design ways to kind of figure this out. And there's kind of 2 prevailing theories why this might be the case. One is that this autoimmune destruction that we previously thought was kind of exocrine specific is not, and that when the immune cells are attacking the endocrine pancreas, they might be killing off some exocrine tissue as well. Another theory is that insulin is this very strong trophic factor. So it's getting pumped out at these very high amounts locally around the islets. And when you've lost those beta cells, after they're killed off, they're no longer producing insulin, you've lost that trophism, and that surrounding exocrine tissue shrinks. And I can say that I'm not going to show it in this talk, but we've found some evidence that this is the case, certainly. We haven't ruled out that there might also be autoimmune destruction of the exocrine pancreas. It's certainly plausible that both of these mechanisms are at play here. So I started to get into this earlier, but we need a way where we can look at pancreas volume across individuals who are 8 years old and weigh 64 pounds, and look at individuals who are 30 years old and weigh 200 pounds. And what we found is that when we look at pancreas size as a function of body weight, there tends to be this kind of semi-linear relationship where the larger your body weight, the larger the pancreas size is. So most of the measurements I'm going to show for the remainder of this talk are going to be a pancreas volume index. That's simply the pancreas volume divided by the subject weight. So it's the pancreas volume in milliliters. We divide that by the subject weight. And when we do that, we find that the pancreas volume index in controls averages about 1. So it's about 1 milliliter of pancreas per 1 kilogram of body weight. Now those individuals who have type 1 diabetes, their pancreas size is significantly smaller. So that pancreas volume index is decreased by about 30%. And that remains when we adjust for age, gender, BMI, any of a number of clinical correlates. So I think this begs the question, well, does the size of the pancreas change? So is somebody born with a large pancreas and they have this autoimmune destruction over time where their pancreas is shrinking over time, and then by the time they're diagnosed with type 1 diabetes, which is kind of the data I've shown you so far, it's smaller? Or are individuals just born with a smaller pancreas and is part of that genetic susceptibility to getting type 1 diabetes stemming from just having a smaller pancreas at birth? So we've started to look at this in a number of different ways. One is we've done longitudinal measurements of individuals right at diagnosis with diabetes, and then following them over the course of the next 5 years or so after that diagnosis. And what we find is that especially in that first year after they're diagnosed, their pancreas size continues to decline. So it speaks to there being some dynamic changes in the pancreas size after diagnosis. On average, the pancreas shrinks by about 10% in that first year after diagnosis. Then it tends to level off over time, but we think it still continues. So when we look at kind of a broader range of diabetes duration, so we recruited individuals who had type 1 diabetes for a decade, 2 decades, 3 decades. What we found is that individuals, as they age, if you have type 1 diabetes, your pancreas volume index, again, your pancreas size divided by your body weight, tends to decline somewhat linearly over time. However, if you don't have type 1 diabetes, your pancreas size tends to stay about the same per your body weight, at least through the 6th and 7th decade of life. There's some literature out there that maybe in the 7th, 8th, 9th decade of life, there is some pancreas atrophy, even in individuals who don't get diabetes. All right, so we kind of spoke about these dynamics of the pancreas size accompanying type 1 diabetes, and I've shown you some information about right here at disease onset. I've shown you even further down this curve after somebody has had diabetes for a year, 5 years, a couple decades. I think the really interesting question is, well, what's happening to pancreas size when people are at risk for type 1 diabetes? So if you're kind of undergoing this autoimmune process, what does your pancreas size look like? So we've worked with the type 1 diabetes trial net, who identifies individuals who are autoantibody positive, who have these high risk factors for getting type 1 diabetes. On average, if you have multiple autoantibodies, so you have 2 or more of these 5 autoantibodies that dkNET or that diabetes trial net currently tests for, you have about a 50% chance of getting the disease in the next 5 years. So we've enrolled over 26 people now who have these multiple autoantibodies in their blood. And what we find is their pancreas size tends to be kind of in the middle. It's a little smaller than those individuals who don't have diabetes, don't have a risk factor for developing type 1 diabetes, but it's a little larger on average than these individuals who are on the right who were just diagnosed with type 1 diabetes. We're starting to parse these out, and what we think is that these individuals who are at kind of the smaller end of the scale are the ones most likely to then progress on to type 1 diabetes, but that kind of research is still in progress here. We're currently following all these individuals longitudinally over time, just like we followed those individuals who were recently diagnosed, because we think those kind of longitudinal dynamics might be really important. All right, so there's this saying that a picture is worth 1,000 words. We think that's certainly true in medical imaging as well. So when we do our images of the pancreas, we capture 30 slices through the abdomen. We certainly get very pretty pictures of the liver. The pancreas will kind of come up here in the middle. It's this kind of bright organ that you kind of see in the middle here. Again, this kind of quasi-banana shaped organ that'll come up through the middle. Now, the data I've shown you so far, we're taking all this data. This is a 3 megabyte image, so a fair amount of data. and we're kind of constricting it all down to this single number, right? This pancreas volume measurement. So if we're going from kind of 3 megabytes of data down to this one number, we're kind of losing 300,000 fold in terms of kind of the pure data, which is, that's a little bit tongue in cheek, but in reality, we think we can find out a lot more from this image than just the size of the pancreas and things that might be important when we think about the changes that are occurring in the pancreas in people who have diabetes. So I think the really easy analogy is to histology, right? If we look at this immunohistochemistry of an islet, we could measure the size of the islet, and that might be important, but certainly people who are looking at islets under the microscope are doing a lot more than that, right? They might be looking at the shape of the islet. They might be able to tell if an islet looks kind of a normal oblong shape or whether it's starting to become irregular in people who are undergoing some autoimmune attack of their islets. In immunohistochemistry, you can get different contrasts by staining the tissue with different antibodies. I think the other important thing is imaging allows you to look at spatial relationships, right? So unlike just a simple number where it's just a number that doesn't tell you about how the different parts spatially might correlate with one another, imaging allows you to do that. So for instance, in this immunohistochemistry here, we can see this kind of spatial relationship, this characteristics of beta cells, alpha cells, delta cells. Well, we can do similar sort of things when we talk about medical imaging. So for instance, I've already kind of touched obliquely on this. The shape of the pancreas we think is different in these people with type 1 diabetes. So these red volumes you see here on the top row is an individual who has type 1 diabetes, again, right at onset. So this is somebody who was just diagnosed with type 1. Hopefully you can appreciate that this red volume is kind of thinner through the body than this individual down here. Here's our kind of characteristic fattened banana shape in our control individual. And we've started to quantify these in some kind of rudimentary ways, at least initially here. We've done things like drawing bounding boxes, right? So found out how much 3D volume this pancreas fills in or comprises, and then also, what proportion of this bounding box is filled with pancreas here. So giving us an idea of kind of the sparsity or solidity of this three-dimensional organ here. We can do something very similar by drawing a bounding convex hull. So imagine kind of taking a balloon that fits around this pancreas and seeing how big that kind of hull is, how much of that hull is comprised of pancreas. We can also draw a bounding ellipsoid. So again, kind of another rudimentary way, but we can basically say the pancreas is kind of elongated, right? What if we draw this ellipsoid around it and quantify those principal axes that kind of define that three-dimensional space? And what we found from this is that all these shape metrics are affected in individuals with type 1 diabetes. So the surface area to volume tends to be increased, right? Because there's more surface area proportional to volume. All these other metrics in terms of kind of the bounding box volume, the percent of the box filled, percent of the hull filled, and these kind of principal axes are all smaller. Now, interestingly, if we take a ratio of the longest to shortest principal axis, it's actually a little bit higher in type 1 diabetes. So what this we think is telling us is that we're able to kind of look at spatial changes in pancreas shape in individuals with type 1 diabetes. And it's not an isotropic loss of pancreas size, right? It's not like the pancreas is just this balloon that we're letting air out. It actually looks like it tends to be this kind of constriction of the pancreas kind of through the middle of the pancreas and less decrease in pancreas size kind of from end to end. Interestingly, when we kind of combine our different metrics, when we combine, say, the pancreas volume index shown in red, this is showing ROC curves for differentiating individuals with and without type 1 diabetes, when we add in there some of these shape metrics, we're better able to characterize these differences in shape, right? So we go from having just this one metric that does a decent job, right? 83% of the time it can tell them apart. But when we add in shape and size, now we're doing, in excess of 90% in terms of capability of characterizing differences between the pancreas in individuals with and without diabetes. All right, so I mentioned when I gave that example of the immunohistochemistry that the contrast can be changed by using different antibodies. Now we can do something similar in MRI. So effectively, when you go to take an MRI image, you have this multitude of different settings that you can alter. And by changing each one, there's basically an infinite number of different combinations you can do, and you can change the contrast based on that. So this is the same individual, basically the same slice through his abdomen. So you can see the pancreas here on this T1 weighted image. Now look over here, this is a T2 weighted image. So we've adjusted the way that we take the image, and you see the contrast changes drastically. So in this T1 weighted image, things that tend to have a high protein content, like the pancreas, show up pretty bright. Things that are liquidy actually show up pretty dark. That's pretty almost diametrically opposed to this T2 weighted image, right? Where things that are liquidy actually show up really bright. So you see the gallbladder here is really bright. The stomach is really bright. Things that are liquidy are really bright. This kind of fluid in the vertebral column is pretty bright here. So really interesting. And the spinal column here is, bright here. So we think that those are not only interesting to be able to adjust this contrast, but we think we can kind of tune the contrast to tell us things about the tissue. And so this is kind of summing up some of those different measurements that we can make that can tell us different things about changes that might be occurring in the pancreas. So for instance, I've already kind of gone over the top one. We can use just a volumetric MRI to give us a sense of the size of the pancreas. We know the size of the pancreas is altered in people who have diabetes. We can also do a technique called diffusion-weighted MRI to tell us about the density of a tissue. Using MRI, we can do a pretty good job of separating the water and the fat tissue, so we can get a sense of how much fat is in a tissue. We can also do things like T1, T2 relaxation maps, magnetization transfer, other techniques that tell us things about protein content, fibrosis, inflammation. The really important thing about all these techniques is we don't have to inject any contrast agent. So we're not doing anything to the person, we're just doing something to the computer, changing the way we acquire the image, changing the way we process the data once we've acquired the image, and then we can get all these data points, all these different kinds of maps at the same time. So we have an individual in the scanner, we take a pretty picture of their pancreas so we can figure out the size and shape, and we can spend the next 30 minutes taking these different techniques and be able to map kind of in three-dimensional space what these different characteristics of the pancreas are. I'm only going to show one example here. This seems to be the one that's probably the most promising for identifying differences that happen in kind of the composition or structure of the pancreas. This is a technique called an apparent diffusion coefficient. It's from a technique called diffusion-weighted MRI. It's actually pretty widely clinically used now, especially in stroke imaging, in oncological imaging. It's a technique that tells about how well water molecules can move around. And so when we look at this color code here, a high ADC value, so these yellow sorts of voxels within the pancreas, mean that the water can really move around. It's a high diffusion value. We don't see a whole lot of them in the control pancreas. I've indicated a couple with the arrowheads here. What this tells us is that the control pancreas is mainly blue and green, so it means the tissue is relatively stable, right? It's relatively impervious to water moving around. Contrast that to the pancreas of this individual who has type 1 diabetes, and we start to see all these yellow spots. So these indicate areas in this individual's pancreas where water molecules can actually move around. They aren't hindered. So we think that this may reflect inflammation that's occurring. We might be actually mapping this inflammation in the pancreas of this individual who's developed type 1 diabetes. Now, these are really complex images, right? We can look at them and there's a multitude of different ways we can start to characterize them, right? We can tell that the pancreas size is smaller in this person with type 1 diabetes. We can tell that the shape is altered, right, between this control and this type 1 pancreas. And we can tell the texture of this diffusion map is different. And texture is kind of what we talk about in the imaging world when we talk about kind of there's different spatial heterogeneity, right? So it's somewhat homogeneous in this control pancreas, but it tends to be pretty spatially heterogeneous in this individual with type 1 diabetes. There's a whole field of kind of ways to characterize these texture differences. There's a whole radiomic field that's trying to kind of quantify these. Kind of more recently, people have tried to use machine learning to do this. And here at UT, we're fortunate to have, well, it previously was the world's fastest academic supercomputer. These things move quickly, and I think we've since been jumped by other academic centers here, but it's plenty fast for what we want to do, which is to try to characterize these differences in the pancreas images of individuals with and without type 1 diabetes. And what we have found is that when we take a deep learning algorithm, we take a UNet, and we basically train it on these different images from people with and without type 1 diabetes, If we show it just the images, so we don't give it any other data, it's already doing a pretty good job at characterizing and classifying the pancreas of individuals with and without diabetes. We're about 75% accurate giving it just images. If we give it not only those images, but we give it some demographic information, we give it some summary imaging metrics, so we tell it, how big of a pancreas did this person have, we give it the person's body weight, sex, age, all those things. Now we're doing a pretty good job. So we're up to about 93% already, and we've actually improved a little bit on there. And the goal, of course, is to get our best job at characterizing these differences between somebody with and without type 1 diabetes. That's kind of I call that kind of the academic problem. The kind of clinical then thing we can move on to is, could we predict it? Once we've built these models, could we use these kind of image classifiers to predict which people at risk for getting the disease look like the pancreases of people who have the disease? And those are the people we think are gonna end up getting type 1. Okay. So I'm going to switch gears here a little bit, and I mentioned that the pancreas is also affected in type 2 diabetes. We've done less work in the field of type 2 diabetes. Most of ours has been primarily in type 1 diabetes. There's a lot of literature out there that shows that the pancreas is markedly smaller in individuals with type 2 diabetes. Now I'm going to kind of give this caveat here that the kind of multimodal imaging signature of the pancreas in type 2 likely differs from type 1. There's a number of people who've kind of started to look at the shape in type 2. And unlike this kind of constriction like we see in type 1, people in the type 2 literature are often seeing these kind of invaginations. They're seeing this kind of irregular border of the pancreas in type 2 diabetes. There's certainly differences in fat content. So these kids we're imaging with type 1 diabetes have little to no fat in their pancreas, where you see a significant amount of fat in the pancreas of individuals with type 2 diabetes. Certainly inflammation we'd expect to be different between type 1 and type 2 diabetes. But when we think about the kind of putative natural history of type 2 diabetes, we think that, it might follow similar trends eventually. Initially, people who develop type 2 diabetes may develop insulin resistance. They may actually have this beta cell proliferation in kind of the early prodrome of the disease. But then over time, those beta cells start to die off as well. So individuals with longer standing type 2 diabetes also start to experience this loss in beta cell mass, again, possibly taking months to years. And that's one of the difficulties in this field, right? It's really difficult to identify or time precipitating events. We look at these individuals at risk for type 1 diabetes who have these autoantibodies. We don't know when they started down that path. It's really hard to kind of define timescales, and it'd be really useful if we had kind of a defined time point where we could say, all right, this was this was some risk factor that we could time, we knew when it occurred, and then we could benchmark our imaging towards that time point to be able to kind of define these are events that happen before and after that time point, or this is an event that happened X amount of time after that time point. So it allows us to get some sort of kind of a temporal marker where we are in that kind of progression of the disease. So that's going to lead me into my dkNET pilot project here, where I've been looking at a study that's hosted on the NADDK Central Repository. It's called the A2ALL study. This is the Adult Living Donor Liver Transplantation study. So this was a study of living donor liver transplantation. We know that about 30% of people who get liver transplant end up developing diabetes. And so my dkNET pilot grant is titled Machine Learning of Abdominal MRI in C and CT in the A2ALL liver transplantation study. And the really great thing about this resource is that there was a whole boatload of imaging done on these individuals. And it wasn't done because people wanted to look at the pancreas. It was because people wanted to look at the liver, right? They wanted to know what the liver looked like before and after transplantation. They wanted to look at liver donors. They wanted to look at liver recipients to get a sense of kind of what the liver looked like and if the liver transplantation was working. Luckily for us, when we kind of look at these axial planes, at these abdominal images that contain the liver, they also have the pancreas. So we're kind of getting the imaging for free there. And thanks to the NIDDK being really nice, good stewards of this data, they have it available to share and have a fair amount of data. So over 1,000 scans, about half MRI scans, about half CT scans, and they're performed at 9 different clinical centers. Now we've started to kind of look at these images, and one of the things that we've found is that the pancreas size tends to be different before and after transplantation in liver transplant recipients. So if we look at an individual who received a liver transplantation 1 week before he got his transplant, I've kind of drawn a red arrow to the pancreas, and I'm going to kind of outline it here as well. So you see it has this kind of barbell shape here in this individual before liver transplantation. 2 months later, he had another MRI. They were really interested in kind of what the liver looked like post-transplantation, but we get the pancreas here as well. And hopefully you can appreciate that the pancreas is markedly smaller in this individual. And we've tried to capture the same kind of axial slice here. You can see the spleen and kidney look similar. Granted, you have kind of this liver transplantation in here as well, so these abdominal organs may have shifted a little, but we can again go through all these different slices and quantify how big the pancreas is. And what we found is that we've now looked at 71 imaging pairs, so that's 71 images performed before liver transplantation, 71 images after. We're again looking at the liver transplant recipients. And what we found is that there's a pretty drastic decline in pancreas volume in these individuals who get liver transplantation. And we hypothesize that this may reflect the altered metabolic demand after liver transplantation and may, in part, reflect this increased risk of developing diabetes after liver transplantation. So we think this is really a kind of an interesting data set because it provides this kind of incident measure where we can know kind of the precipitating event, right? We have imaging before and after this precipitating event, and we're kind of at work now, both performing more measurements in this data set. We're made it through about half of this data set so far. And then we're correlating this with different clinical measurements that are also contained in the database. All right. Unfortunately, making these measurements is really time consuming. So far, we've analyzed 247 images from the A2ALL study. What we have to do this manually is actually go through every single axial slice and manually kind of draw the contours of the pancreas, right? It's really time consuming. It's tedious. I'm looping through here through the movie, and you'll see that the kind of yellow is the pancreas. You can hopefully appreciate there's pancreas present on many different slices. On average, it's probably about 20 slices that have pancreas on them. So every single individual that we analyze, we have to go through at least 20 slices. Depending on what imaging modality it was and how thin the slices were, some of them were having to go through upwards of 40 slices to accurately quantify the pancreas. And this is time consuming. It's a lot of work for this study. It's too much work if we're thinking about making this into a tool that we could apply to really huge data sets. And so one of the things I've been working on, and many others in kind of the medical imaging field have been working on now for decades, is, well, can we train computer algorithms to automatically segment different organs? And these have really taken off, I would say, within the last 5 or 10 years, between advancements in deep learning, UNets, and what we've found is that they can do a pretty good job now. So we've taken our data sets where we've had a radiologist go through, manually segment out the pancreas, and for some individuals with type 1 diabetes, some control individuals, shown in red here, the kind of segmentation of the pancreas. Note the drastic difference in the size and the shape of the pancreas here. So we train this algorithm on many of these different data sets, and we think it's important to train the data sets with pancreases that are both derived from people with diabetes and without, so you get kind of this full representation of the data here. And when we do that and then show the algorithm a pancreas that's never seen before, ask it to segment the pancreas, it does a really good job. We get a Dice coefficient in the 0.8. And pretty good overlaying of these two. We tend to see a little bit of disagreement between the human reader and the segmentation at kind of the margins of the pancreas. And one of the things that I've found is that if I really kind of zoom in and look at the pancreas, sometimes the automatic segmentation almost looks better than the manual segmentation, right? So our human reader is kind of limited by how closely she can go around and accurately define those kind of very small changes in image contrast. Our machine learning algorithm, I think at times, is actually outperforming our gold standard. So it's kind of interesting to think of ways to accurately kind of quantify those differences. When we look at kind of our measurements of the pancreas size in people with and without type 1 diabetes, we see very good agreement between our manual pancreas volume, so that's the radiologist-derived measurement, and these automatic pancreas volume measurements. So now we're using artificial intelligence, machine learning to be able to hopefully automatically make these measurements of pancreas size. Now we're thinking about turning our attention to this A2AL study. We have very different image contrasts. As I mentioned, we have about half our data set comprised of CT images, about half the data set comprised of MR images. You see drastically different image contrast here, right? Pancreas looks very different, the borders look very different. So we're looking at ways now of trying to make a pancreas segmentation algorithm that can take into account these differences in CT and MR images and perhaps make two different models, look at the image, is able to derive which one is CT or MR and apply the correct model. So I've done a fair amount of thinking about these kind of multi-site trials. So we've actually, for the past 5 years or so, been developing this consortium we call the Multi-Site Assessment of the Pancreas and Type 1 Diabetes, or MAP-T1D. And it's a consortium that's trying to make this pancreas imaging reproducible across different sites. So I'm working with diabetes centers at Vanderbilt, Chicago, Colorado, here in Austin, as well as a group in Melbourne. And what we know, those who kind of do medical imaging and operate on different MRI machines, we know that with those kind of multitude of different settings you can change on an MRI machine, the contrast is always going to look a little bit different. Even if you try to set everything exactly the same, it's going to look a little different. This is my pancreas scanned on a Philips MRI machine at Vanderbilt on the top, a Siemens MRI machine here in Texas, and you can see there's a difference between these two, even though we've tried to set everything we can the same. So in order to try to kind of quantify these site differences, we did a reproducibility study where we sent phantoms with known MRI properties to these different sites. And then we actually had 5 different individuals travel around the country. So they went to these different MRI sites, had an MRI of their pancreas. And what we found is that we kind of standardized these measurements. We can do them reproducibly. We think these kind of standardizations are going to be really important for kind of moving to multi-site clinical trials. People in the kind of neuroimaging field who are years ahead of us in pancreas imaging have really already proven the need for this kind of site standardization. And my hope is that in pancreas imaging, we don't make the same mistake that we kind of start out when we're making prospective measurements of making them as similarly as we can. So I think I'm just out of time here, so I'm going to conclude briefly by just saying we think conclusively the pancreas displays reduction in size during the development of diabetes. It's probably smaller before people get diabetes as well. We think this A2AL study is really interesting because we found that the pancreas declines in size after liver transplantation. We think it's important because we can look, kind of before and after an event and see that this event actually triggers a change in the pancreas. MRI, the picture tells 1,000 words here. MRI can characterize pancreas size, which is important, but pancreas shape, pancreas structure, pancreas composition all seem to be affected in type 1 diabetes, and deep learning is really kind of uniquely suited to be able to characterize these different changes as well as segment the pancreas. So I'm going to end here by thanking all my collaborators on the project. The JDRF was really instrumental in funding these initial studies. We're currently funded by NIDDK, the Beatson Foundation, and the dkNET pilot project to continue this work, and I'm very grateful for that. And so hopefully at this time I can take some questions, and hopefully I've convinced you that there's a role for medical imaging in diabetes, and we might be able to help inform our understanding of the disease. Thank you.
Thank you, Jack, for the presentation. So now is our Q&A session. If you have any question, you could unmute yourself to ask or use chat function in the Zoom. I see there is one question in the chat. Given the high cost of MRI, where do you envision pancreas MRI being used clinically in diabetes care?
Right, so that's something I always hear, right? And we're not going to order an MRI for everybody who gets type 1 diabetes, right? That's not economically viable. It doesn't make sense. It's not going to tell us things for everyone. I think that the place where MRI may be useful is things like clinical trials, right? Where you're doing a large clinical trial, you need to stratify patient populations. You want to perhaps track changes that might be occurring to the pancreas. We don't have a whole lot of data in the type 1 field that shows us that changes in the pancreas are tracking therapeutic efficacy. But there's a study by Roy Taylor in the type 2 diabetes community that showed that successful therapy actually induces changes in the pancreas before they're sensed metabolically. So I think in the kind of clinical trial world, there'll be a role for that. I also think that when we start to think about classifying these different endotypes of diabetes and characterizing them kind of much more finely than just type 1 versus type 2, right? We're going to think there's going to be multiple subtypes of the disease, and there might be a role for pancreas imaging and kind of characterizing these different endotypes as well.
Okay, thank you. I think there's a question from John Cadiz, Dr. John Cadiz.
Hey, Jack. I've learned from forums like these, radiologists have said that during the course of a single day, the pancreas can change volumes several times. So you can get a lot of change within one day. So the data that you showed with regard to the your conclusions with the liver studies, and even with the pancreas study, how do you address that? Your slide on the microstructure, which if I'm understanding you correctly, you're sort of arguing that a T1D pancreas is more susceptible to water permeability than a control pancreas. Please correct me if I'm wrong. That would suggest that it might even be more prone to error if you've got something other than disease affecting it, perhaps. So I'd like to hear your thoughts on that.
Yeah, and I think that's one of the things that's difficult, right? And I talk to my friends who are imaging the brain, right, which is kind of a contained organ where you're not dealing with motion. And so in many ways, that can be an easier thing to image. And pancreas is difficult because it's moving around from respiratory motion. And I think that there are probably changes that occur over time, over the course of the day. We've tried to look at that in a systematic manner, and we do see minute changes in the pancreas, but we don't see large changes. We probably see changes on the order of maybe 10% over the course of the day, and we've tried to induce changes, right? We're doing a study right now where we're giving somebody a feeding challenge and then looking and seeing if their pancreas changes in response to that feeding. We don't see a big change there. So in our hands, it's not something we've seen huge changes in. I would argue, though, that if we're making our measurements in the face of these kind of drastic changes, right, we're probably going to get better discrimination. We're probably going to do a better job of characterizing these changes if we can figure out what induces these changes and then account for those, right? Say, hey, if we need to make somebody fast for 6 hours before we do the MRI, or we need to make sure they're, a multitude of, we need to image them at the same point in the day. So there's something that came out of the UK Biobank that showed diurnal changes in pancreas volume over the course of the day. Do we always need to be imaging somebody at noon or have some kind of factor that accounts for that? I think it's all interesting questions. I think that it's going to complicate things, certainly, but I think there are things that need to be investigated, because I think, to me, all those complications say to me that, well, if we're already able to show these differences now in the face of all these challenges and all these covariates that are probably going to be obscuring our results, hey, if we figure out what those covariates are and we account for them in a controlled manner, we'll even be able to do a better job. That's my understanding. That's maybe a glasses half full imaging scientist sort of answer there, but I hope that answers your question.
Thank you.
Thank you, John. So our next question is coming from the Albert Wang.
Hi, Jack. Great talk. And I have 2 questions. One question is somewhat related to John's. So these people had liver transplants. So I think you have mentioned in the past that the volumetric measurement by MRI is, complicated by the fact that pancreas is easily deformed. So then I was wondering whether it will be useful to use like a control group that, receive other kinds of transplant in the abdomen, like, I don't know, other, intestine, transplant or something to sort of account for that. So that's one question. The other question is, I don't know if you saw a paper that just came out maybe like a week ago on a neural network of machine learning on UK Biobank.
The Watson group.
I forgot the name of the group, but then they call this abdominal age calculation, and they looked at liver and pancreas MRI. So, and then they use these like images from this biobank. So I was wondering whether there's an opportunity to use these type of, past data that's already been collected, and then they come with, the information about the medical history to learn, just agnostically. Because what you're presenting is that we already pick something that we're gonna measure, like we're measuring volume, we're measuring the coefficient of diffusion of water. Whereas I think this paper was just throw the data at the computer without prescribing what they're measuring. So I was wondering whether there's a role for machine learning to take the data and look at it that way to look for correlations with diabetes.
I think that's a great question. Nice to see you again, Albert. I'll take that second question there first, perhaps. And I think I was thinking of a different paper. I'm not sure that I've seen this one, but in general, I think that there is a huge role for AI in classification. And I think the nice thing about AI is it's not subject to the biases we have, right? So I'm coming in there thinking that this is a pancreas problem, and we're looking specifically at the pancreas. But if you take a AI and you feed it in a bunch of these images, it's going to look at the holistic image, right? And it might detect, hey, there's a change in the spleen or the kidney that also enters into it. And there's this whole field of explainable AI now, where AI used to be kind of this black box, and it would spit out, all right, these are the two groups, but we can't tell you why we made this decision. Now, explainable AI is saying, all right, these are the two groups, and these are the image features that we detected. So, I could think about kind of hypothesis generating sort of ideas of kind of using AI to define imaging features beyond the pancreas that maybe are different between the two, right? So I think hopefully that addresses the second question there. And I think the first question was about kind of deformation of the pancreas. And that there's certainly possible that the pancreas may be altered after liver transplantation. So I guess the thought is that the liver is in there and everything's getting squished up, right? The point that I would make that argues against that, we've done a little imaging in pregnant women. So these are women who are, everything is squished up in the abdominal organs, and the pancreas actually grows a little bit despite that constriction there. So I think that argues against the pancreas being distended like that. And we've also looked at people before and after a large meal, where you see the stomach going from empty to the stomach taking up half the abdomen, and you don't see that inducing a change in the pancreas. So I think when things get squished around, other things get squished and moved out of place. But I don't think that the pressures that are in there are strong enough to really push the pancreas in. That's we don't have anything other than this kind of very cursory sorts of forays into that. But I think it argues against that.
Great, thank you.
There is one question in the chat. Are there new MRI techniques that may be applied to assay the pancreas?
There are every day there's a new MRI technique, right? So there's our big ISMRM is our MRI conference, and there'll be sessions and sessions of everybody who has a new MRI technique, and they all have their favorite new acronym, they've dubbed it. And so there are a lot of techniques out there that I think would specifically be useful for pancreas imaging. Elastography, I think, would be really interesting. I think there's a couple of groups out there looking at elastography, and this actually kind of dovetails a little with what Albert said. So for MR elastography, you're actually applying this kind of shear wave to the pancreas and seeing how it deforms, and it tells you a little bit about the stiffness of an organ. So people use it a lot for things like liver cirrhosis and things like that. And I think it would be useful for looking at the pancreas. It's not something we've looked at yet, but I would like to in the future.
So I'm not sure if this was asked already, but I'm wondering, would it be worth the effort to label all organs in this A to all data set? I mean, kind of as a community effort. And I mean, this would enable many people to look even at correlated changes in organ form or size or so.
Yeah, I do think that would be really useful. So, the idea would be to segment out the kidney, the liver, the pancreas, everything. One of the problems we'll run into with this A2all study, and which I think is problematic for any of these imaging studies, is that, not only is the imaging device different, but the way the image is prescribed is a little bit different. So oftentimes we'll see, you know, the organ or the MRI or CT centered, you know, pretty high up. So we'll even miss, you know, parts of the bottom of the pancreas, or sometimes it's too low. So we'll miss, you know, perhaps the tail of the pancreas there. And I'd have to think about what organs are in most of the data sets. We'd probably be able to get majority of the kidney, but we'd probably be missing the kidney in some of the organs as well. It's one of these things with, you know, retrospective studies. It's great because we get the data for free. It's already been taken. It's out there, but you know, I would love to go back to the MRI tech and say, oh, could you just add like 5 more slices to the bottom of this scan so that we can get the whole pancreas, and we can't for the pancreas. And my thought would be for a lot of the abdominal organs, we're also going to get cut off just because they're always centered on the liver there, and they don't tend to take a lot of images on either slide. We're just fortunate that typically the pancreas is kind of in that same plane as the transplanted liver.
Mm-hmm.
A great idea, though.
Yeah.
I love that idea.
Yeah, I mean, the idea is actually from, I mean, in machine learning, ImageNet, that was at some point by Stanford professor Fei Li. She just made a huge database of labeled images, and this made the whole field of computer vision take off. So this might be it was a really good investment in that sense. I mean, it was a lot of effort to label these. So maybe this could actually be quite helpful with all the constraints that you mentioned. I agree. This is then a bit of a thing. Maybe one more, if that's okay, with on the calibration on just to make the different machines make the images look similar. I wonder. I think this is also there. I mean, there must be calibration sources or something from the supplier. Have you looked into this? That Siemens or Philips, that they provide actual devices because they have to demonstrate, okay, this machine works according to specs.
They do. Oftentimes those are more on the qualitative aspects, or they'll be on the, you know, so most people who are buying an MRI machine are doing it because they want to get a pretty picture, right? So often those kind of benchmarks for the MRI systems will be you know, a kind of phantom that has kind of a spatial grid, so you can pick out, you know, the spatial resolution of the image is this, right? And you can get kind of those sorts of measurements. In terms of kind of the more research oriented stuff, so the things that we're looking at, like, you know, quantifying diffusion-weighted MRI, making quantitative T1, T2 maps, typically the vendors are not into that as much, and there I think there's a movement to make imaging and MR particularly more quantitative, but most of those kind of phantoms that I talked about, those aren't made by the vendors. They are kind of third-party people who are making these gels and polymers and stuff with prescribed MR fields. But I think there's a push to get the vendors to do that, but it's just not there yet because they're, you know, most of their customer base doesn't care about the numbers. They care about a pretty picture, so the radiologist can make their diagnosis, right?
Okay, interesting. Good. Yeah, I mean, great effort to look into this as part of your study. It's good.
Yeah.
Thanks.